So what are the light reactions? We've moved to the light-dependent reactions, the part that requires light in order to occur directly. We're really talking about two major photosystem complexes and a couple of accessory proteins. All right. So the first photosystem is actually called Photosystem 2, and it's called Photosystem 2 because it was described or discovered second. The second photosystem is called Photosystem 1. You can eliminate all this issue by calling Photosystem 2 P680 and Photosystem 1 P700. P680 is the wavelength of light energy that the reaction center of Photosystem 2 absorbs light energy at. And P700 is the wavelength of light in nanometers, 700 nanometers, that the reaction center of Photosystem 1 absorbs light. All right. The photosystems are, just as the electron transport chain is in cellular respiration, is embedded in the membrane, although the membrane here is not going to be the inner mitochondrial membrane. The membrane here is called the thylakoid membrane. All right. Outside the thylakoid membrane is going to be the stroma, and inside is going to be the thylakoid space. All right. Now remember, we said that creating light energy, or creating energy from light, is a process that really is driven by the use of electrons, just as in cellular respiration. But the electrons here are not going to come from organic molecules, because that's what we're trying to make here. All right. The electrons are going to come from the splitting of water. All right. So generally, light energy enters through the leaves, through the cell, into the chloroplast, and basically gets received by the various pigments of what we call a photosystem, or a light harvesting complex. All right, which each of these photosystems has. Again, redox reactions are going to transfer electrons from a high energy state to a lower energy state. We're going to pump hydrogen ions across the membrane. All right, before the electron gets re-excited at a different energy level, P700, 700 nanometers this time, and to then get changed into another energy state where we have a reduction going on at the end here. But generally, the processes are the same, except the membranes which they're occurring, the organelles which they're occurring, and there's going to be a few major differences about where we get our electrons from and where electrons are going to go. All right. Specifically, the light harvesting complex is a complex all around here. It's embedded within this protein of many different molecules and atoms that are designed to transfer incoming light energy from one place to the next until it gets to the center, what we call the reaction center. The important thing to know, the photosystem is the entire complex. All right. Now the photosystem includes the light harvesting reactions, all right, the light harvesting complex, and it includes the reaction center, the photosystem. Okay. Now the reaction center is a pair of chlorophyll A molecules. All right. So all of the pigments that surround a photosystem are really a mix of chlorophyll A and B and carotenoids and different types of accessory pigments that are designed to take in as much light energy as possible, absorb as many photons at different wavelengths. This energy gets resonated and transferred and absorbed around the photosystem to different pigments until it reaches the reaction center chlorophyll molecules. All right. These two reaction center chlorophyll molecules lose their electrons to a part of the redox reactions that go on in the photosystem, and then that energy splits water and takes electrons one by one from the hydrogens of the water molecule. All right. Now, photosystem 2 has a reaction center called P680 because that's where it absorbs the most light at, raising its energy state. And photosystem 1 is also called P700 because that's the energy level that it works at. Okay. I actually don't know why they work at such different levels. All right. But it just makes sense that the electrons in photosystem 2, by the time they get there, are likely to be less energized because their energy has been used to do work, and it takes a lower wavelength or a longer wavelength, lower energy wavelength in order to excite them to another level again. All right, that'd be something to research on your own if you're really interested on why one is actually has a reaction center at 680 nanometers and why another one has a reaction center at 700 nanometers wavelength. All right. This is just a more complex and a little more descriptive diagram of all the redox reactions that are going on at one time. All right. The redox reactions, again, are designed to transfer electrons from one place to another so that we can use these electrons, number one, to reduce NADP plus to NADPH, and also to use these electrons to pump hydrogens across the membrane. All right. Again, we're using the electrons to pump and release hydrogens across the membrane, all right, into the thylakoid space, all right, by which ATP synthase will then use this proton gradient to make ATP. Right? So now what we end up with is lots of ATP from ATP synthase and lots of reduced NADPH to carry electrons to the Calvin cycle, or the light-independent reactions. All right. So some important things to know is the difference between the reaction center and a light harvesting complex, also called antenna pigments. All right. And where the electrons are coming from, where is things happening? All right. What is the point of our mobile carriers? All right. What is the point of our plastoquinone cytochrome B6 complex? What is the point of this complex here? All right. And a little bit about how these two photosystems differ from one another. Photosystem 1, P700, is actually the photosystem that reduces NADP plus to NADPH. So it's really the one that provides us with our mobile electron carrier so that we can have reductive power later on. All right. But all of this stuff is being energized by photons coming from light, which is really important. All right. There's just another example of how extremely complex all these proteins are. This is photosystem 2. This is the cytochrome complex, and you can see this is a channel to allow hydrogen ions to come through using the energy from electrons. We also allow hydrogen ions to come through as the mobile carriers move across from one place to another. And also we have our photosystem 1 here with our ferredoxin. Ferredoxin is basically the molecule that is going to be reduced, and it's going to help us reduce NADP plus to NADPH using those electrons taken from initially what would be water. Remember, in cellular respiration, ATP synthase is taking an electron gradient that is higher in the mitochondrial inner mitochondrial membrane, all right, in the intermembrane space, I mean, and the electrons are flowing into the mitochondrial matrix. On the other hand, in photosynthesis, your hydrogen ion gradient is being created in the thylakoid space, and the hydrogen ions are being allowed to flow through the ATP synthase into the stroma. And that's important because ATP, and NADPH are going to be used for the Calvin cycle, a reductive process that also occurs in the stroma. All right. So again, light energy energizes pigments that transfer energy to the reaction center where electrons are being taken from the splitting of water. Hydrogen ions and oxygen are created. All right. We have mobile carriers here, and we have cytochrome B here, which is also going to help pump hydrogens across the membrane. And we have our photosystem 1 here, which again takes the electron. It doesn't split water because we already have the electrons, but light energy is transferred from the light harvesting complex to the reaction center where electrons are transferred via redox reactions to ferredoxin, where we can make NADPH from NADP+, and we can then make ATP from the hydrogen ion gradient and ATP synthase. All right. So this process doesn't differ much, except where it happens, on the membrane it happens, where we get our electrons, where our energy is going to come from. So it'd be helpful to focus on the differences between the two. Just another perspective here, the light reactions, these reactions here are occurring on the thylakoid membrane. We are going to be pumping hydrogen ions into the thylakoid space, allowing them to go into the stroma. The important parts of the light reactions are to create ATP and to reduce NADP plus to NADPH using electrons, light energy, water, creating oxygen as a byproduct. Now there's one more thing that can happen in many organisms. It's called cyclic electron flow. All right. Remember we talked about feedback and the things that happen if the cell realizes that it doesn't actually need something. Well, let's say that the cell doesn't need to make any more sugar. All right. For some reason, it's not growing. It doesn't need to make cellulose. It's not able to make starch. Or for some reason, it just doesn't need for the Calvin cycle to occur. The Calvin cycle's point or purpose is to create sugars, right? Well, let's say the cell doesn't need to create sugars. Maybe NADPH is building up. If NADPH builds up, then the electrons are not going to be transferred onto ferredoxin. All right. Well, actually, yes, the electrons will be transferred onto ferredoxin, but ferredoxin, instead of taking the electrons to NADP+, it will take the electrons back through the cytochrome B6 complex, whereby the electrons will then go back to plastiquinone and release them again to photosystem 1. So here, we're taking electrons and we're cycling the electrons through, all right, which means that we can create that hydrogen ion gradient necessary to create ATP. All right, and we can also reuse our electrons, and all we need is light energy to continuously excite the accessory pigments and P700, or the reaction center. All right, so cyclic electron flow is a way for organisms to actually react to not needing sugars, but still needing ATP. All right, why would an organism make sugar and then break it down for energy if it could just make energy right away? So cyclic electron flow is a way to make ATP directly and not have to go through the process of the Calvin cycle in order to make sugars and then send those sugars to the mitochondria where then energy can be made. So it's we're saving a bunch of steps by being able to use cyclic electron flow. It is also thought that photosystem 1 was actually the first photosystem to actually exist because it's found in many very, very simple organisms, and that cyclic electron flow might have been the only method of making energy for many very simple organisms in evolutionary history. So overall, what's going on here? We have the organelle called the chloroplast in the plant cell. It's a double membrane cell. The chloroplast houses these things called thylakoids, which a stack of them are called grana. Many stacks are called grana. These thylakoids have embedded on them photosystem 1 and photosystem 2 and some other membrane proteins and mobile carriers that are designed to make ATP and NADPH from NADP plus and NADP using electrons from water, all right, creating oxygen as a byproduct and using the energy from light in order to excite the photosystems to break water and move electrons, create a hydrogen ion gradient, and then make ATP from ATP synthase from the hydrogen ion gradient, and reduce NADPH using the electrons that we took from water originally that came from the chlorophyll A molecules.